This video is on the cell cycle. The cell cycle is a repeating series of cell growth and division. And why do cells divide, first of all? Let's start with that. So cells divide by mitosis for a number of reasons. First, they divide for growth. For example, if you start with a zygote, which is a fertilized egg, right? It's got to grow into an organism, which means we need to add more cells. 2, cells grow when organisms need to be repaired. For example, if there are cuts, if you have a broken bone, if you have damaged tissue, you need to make new cells to repair them, those old cells or the cells that have died. 3, and this kind of fits into these other ones as well, we need to increase surface area to volume ratio. This helps with transport efficiency. Because remember, small cells are more efficient. So if you have one large cell and you divide it into multiple smaller cells, this increases transport efficiency and helps the cell maintain homeostasis. So when you guys were freshmen, you probably talked a little bit about the cell cycle. But when you talked about the cell cycle, you talked about it as a series of steps. And you probably divided it up into interphase, mitosis, and cytokinesis. We'll do that first, and then we'll move into how the cell actually really thinks about its division. So again, when we think about the cell cycle, in kind of a classic freshman biology way to teach it, we think first about interphase. An interphase is typically considered the longest phase of the cell cycle. An interphase has a couple major components. The first component is G1. This is when the cell grows. And this is when the organelles, like the mitochondria, replicate. Next, we have S. S stands for synthesis. This is when the DNA is copied. And at this point, the DNA is in chromatin form. And then the third part of interphase is G2. At this point, we're really proofreading and we're repairing DNA. So G1, S, and G2 all fit into interphase, and we're really preparing the cell to divide, right? So you have G1 here, and you see the cells growing. We have our unreplicated chromosomes here. Then we get into S, where the DNA replicates itself. Then we get into G2, where the DNA is checked to make sure it's correct. And this is all part of interphase. The second part of the cell cycle is mitosis. Mitosis is a much shorter step, and this is when the nuclear material divides, when the DNA divides. Mitosis is the division of the nuclear material, aka division of the DNA. And mitosis really has 4 major steps itself. So prophase is the first step. This is kind of our prep step. This is when our chromatin, our long uncoiled DNA, really condenses into our chromosomes, our DNA suitcase. Then we have metaphase. This is when the chromosomes line up in the middle of the cell. So prophase prep, metaphase chromosomes are in the middle of the cell at the equatorial plate. Then we have prophase, metaphase. Then we have anaphase. This is when the chromosomes move apart, anaphase apart. And then we have telophase. Telophase is when you have 2 nuclei. That's when the new nuclear membrane have reformed and you have two nuclei there. So that kind of takes you to the end of mitosis, the division of the nuclear material. So mitosis comes right after interphase. Prophase, metaphase, anaphase, and telophase. Then, kind of at the end of mitosis, we have this, sometimes we classify it as a third step. Sometimes it's kind of the end of mitosis. We have cytokinesis. If mitosis is when the nuclear material divides, so at the end of mitosis, you kind of have a cell with two nuclei in it, right, in telophase. Cytokinesis is when we finally have that split between the cytoplasms of the two cells. So cytokinesis is just the division of the cytoplasm that happens after the nuclear material has split. There's many different mechanisms where this can happen. In animal cells, something called a cleavage furrow occurs, where the animal cells are being pulled apart, and it kind of looks like, if you pull like gum apart, it's thinner and thinner and thinner, and then there's a split between the cytoplasm of the two cells, and then the cell membranes kind of are completely formed around each animal cell. In plant cells, you have a cell plate, right, which then becomes more and more defined, and you have more material put there until your cell wall forms. So this is kind of like classic cell cycle, okay, the way we think about it freshman year. We need to add a couple things in, and I also want to talk about this from the perspective of the cell itself. If you notice on this diagram, this is the picture of the cell cycle. On this diagram, you might see that something called G0. A lot of times students think, okay, interphase is the longest phase of the cell cycle. This could go on for years. When the cell's not dividing, it's an interphase. But that's actually not true, okay? When the cell starts to divide, this whole process is fairly continuous for mitosis. We'll talk about meiosis later. But a cell can either be dividing or it can be doing its job. So an intestinal cell can either be dividing to make more intestinal cell, or it can be doing its job in the intestines, digesting food, absorbing nutrients, whatever it's doing, okay? If a cell has left the cell cycle to do its job, we say it's in G0, which is separate from interphase. It's outside of the cell cycle. So G0 here is when the cell has left the cell cycle and it's not dividing. This is where the phase the cell is in when it's doing its job. When it's making proteins, when it's sending signals, when it's whatever it's doing, okay? So G0 is outside of the cell cycle. Let's get back into the cell cycle. So again, like I said, this is how we think about it freshman year. Interphase, a time of growth and DNA replication. Mitosis, a time of nuclear division of the DNA. Cytokinesis, a division of the cytoplasm. That's not really how the cell thinks about it, though. The cell wants to make sure that these steps occur correctly before we move into this step, okay? So for instance, if the DNA has not copied itself correctly, the cell will proofread the DNA. And if the DNA has not copied itself correctly, the cell will not allow itself to go into mitosis, okay? In mitosis, the main sticking point is in metaphase and anaphase. If the chromosomes don't line up correctly at the equatorial plate in metaphase, then the cell won't move into anaphase. And I say won't, obviously there are exceptions to the rule. There are things that happen, there are mutations, but I'm talking about in a healthy cell, how does the cell cycle work? So the move from metaphase to anaphase won't occur if the chromosomes haven't lined up correctly. So when we think about mitosis now, we're really going to focus on cell cycle control. How does the cell make sure all of these steps have occurred correctly before it moves into the next step? Okay, and so instead of thinking about interphase, mitosis, and cytokinesis, I'm going to divide up the cell cycle a little bit differently. I'm going to use this diagram up here to do that. In this diagram, you see a diploid cell. Remember, diploid cells are 2N, meaning they have 2 copies of each chromosome. So here are 2 copies of this chromosome right here. At the end of the process, you start with one diploid cell in mitosis, and at the end of the process, you end up with 2 diploid cells. Okay? So the first thing that has to happen is we have to copy the DNA to make sure that the end result is 2 cells with the same number of chromosomes as the original cell. So when I'm actually dividing this up in what the cell thinks about, the first step I'm really thinking about is DNA replication right here. So for me, this is the first important thing that really happens in the cell cycle. Okay, the replication of DNA in S phase of interphase. So first step here is replication, right? DNA copies itself, and this occurs during S phase of interphase. This is kind of the first checkpoint. Did this happen correctly? If yes, we can move on. If no, we don't move on. Okay, so replication. The second checkpoint is a checkpoint that happens during this process of mitosis right here. So here's prophase, metaphase, anaphase, telophase, and cytokinesis here. The second checkpoint is one that happens between metaphase and anaphase here. This second checkpoint here is checking on alignment of the chromosomes. Alignment is, are the chromosomes have the chromosomes lined up appropriately at the equatorial plate during metaphase of mitosis. So that's the second checkpoint here. The third checkpoint is have the chromosomes separated correctly. That also occurs during mitosis. Okay, and kind of mitosis to cytokinesis here. So here's our third checkpoint right here. I'm going to write it over here because I'm running out of space. Our third checkpoint is separation. Separation of the chromosomes during anaphase of mitosis. So once again, these are kind of the checkpoints we're going to talk about in the next video, which is about how the cell controls the cell cycle control. I would be remiss, however, if I didn't briefly mention what happens in prokaryotes, because obviously up there we're talking about eukaryotes. In prokaryotes, and again with prokaryotes, we're talking about bacteria. Bacteria divide in a process known as binary fission. So if this is the DNA right here, prokaryotes are a little different because we know eukaryotes have linear strands of DNA, okay, that are replicated, and then we have to make sure all those linear strands get to all the different cells. For example, humans have 46 chromosomes. In mitosis, we want to make sure every single cell has 46 chromosomes, right? 46 strands of DNA. Bacteria, you have the main chromosome and then maybe some little plasmids, but really we have a single loop of DNA. And replication starts at one point. So the point on the DNA where replication starts is called the origin of replication. And the DNA just replicates all the way around. And then instead of having one single chromosome, you have 2 copies of that chromosome. So that when the cell divides, each new bacterial cell has a copy of that one loop of DNA. So as the cell, I guess I should say, as the DNA replicates, the cell grows and divides. And you can actually see this over here, right? Binary fission. Here's the original loop of DNA right here. It starts to be replicated, right? Then as it's replicated, the cell grows, and oftentimes that bacterial chromosome kind of anchors itself on part of the bacteria. And so as the cell grows, the two chromosomes that are identical to each other move apart. And again, you have the cell wall forming. And then in the end, you have two cells with identical DNA. And this is a little bit more straightforward because you only have to separate this one chromosome, not multiple chromosomes. That ends this video. In the next video, we'll talk more about eukaryotic gene regulation, although bacteria regulate their replication as well.